Hello and welcome back to educator.com. Today's lesson will be on gene regulation in eukaryotes. So first we'll talk about transcriptional regulation, and we'll also talk about the regulation of translation. Finally, we'll talk just a little bit on epigenetics. So let's first talk about our eukaryotic transcriptional regulators. Mainly, we're going to talk about our transcription factors, and there are many different types of transcription factors, usually characterized by the specific domain by which they bind DNA. So we have homeodomain proteins, we have zinc finger proteins, we have leucine zippers, helix loop helix proteins, as well as HMG proteins. HMG just stands for high mobility group. We also have insulator proteins, in which these are proteins that bind between the enhancer and the promoter in a gene, and they suppress transcription. So if we remember, enhancers are usually far upstream of where the core gene element itself starts. Up here are transcription factors. They can be either enhancing or repressive. And compared with our prokaryotes, transcriptional regulation and regulation in general for eukaryotes is much more complex. Usually there are more regulators and longer length of regulatory sequence in the DNA. So let's first start off with an example of transcriptional regulation. So we have Gal4 and Gal1. Now Gal4 is a protein and Gal1 is a DNA sequence. Now we're looking at this in the yeast Saccharomyces cerevisiae. Now the Gal4 protein will bind to a site upstream of the Gal1 gene. And this is going to increase transcription by Gal1, right, the Gal1 gene, by about 1,000-fold. So what this would look like, all right, Gal4 binds as a dimer. And what we see here, if we draw this out, all right, so guys, right here, this arrow, that's a shorthand form of saying that's where a promoter is, and Gal1, that's our gene. All right, this right here is a UAS, or an upstream activating sequence, and there are 4 sites here at which the Gal4 protein will bind, and it will bind as a dimer, okay? It'll bind as a dimer, and between the UAS and the Gal1 gene is about 275 base pairs. All right, so the binding upstream over here will affect how this gene is transcribed. And we can increase, okay, so these are Gal4s, remember? So these are G4s. This can cause an increase of transcription about 1,000-fold. All right, so if for example, you were supposed to only make 1 mRNA, or in a certain period of time, you make 1,000 times more than that. All right. So let's talk about our locus control regions. All right, and first off, we're going to draw this out. So I guess we'll use red. Okay. So here is our locus control region. Okay. Then we have our globin genes. Okay. Oop, that should be gamma. Sorry, not delta. This is delta. This one is beta. Okay. Now, right here is our cluster of globin genes. Okay, so we have locus control regions, all right? And locus control regions are just long-range regulatory elements that enhance expression of our linked genes, all right? And this helps function in a copy number dependent manner and is actually tissue specific. So for this one, for example, this allows this locus control region. Okay, this allows ordered transcription of downstream genes. And what that's really saying is that we have selective expression of our beta globin genes in our erythroid cells. So this is an example talking about our selective. Okay, erythroid cells are also known as red blood cells. So our locus control regions allow for ordered transcription of downstream genes. So here's our locus control region. Okay, and here are our downstream genes. We have our 5 globin genes. We have epsilon, we have gamma G, gamma A, delta, and beta. And during human development, all 5 genes are transcribed sequentially, meaning first epsilon all the way through to beta. Now, what happens here is that we have euchromatin position. If we remember correctly, euchromatin is opened, more transcriptionally available. Heterochromatin is closed, less transcriptionally available. So euchromatin position will change and heterochromatin formation, reformation will follow behind. So this is something that I want you to just keep in mind because we will be talking about epigenetics later and how euchromatin and heterochromatin positioning affects the transcription, translation of genes, as well as, the transcription of genes, as well as the replication of DNA sequence. Okay, so here are locus control regions. This is one example. Okay, now let's move on to signal transduction. All right, so signal transduction occurs when we have an extracellular signaling molecule activating a specific receptor located on the membrane of the cell. Okay, now this is, so if I draw it out, here's a cell. Okay, here might be a receptor, and here is our extracellular signaling molecule. Okay, so when this molecule binds to the receptor, it's going to trigger a chain of events, okay, inside the cell that creates some sort of a response. And that response is going to be different based on which pathway is being activated. Okay, so some of our examples that we'll talk about today are NF-kappa B, the JAK-STAT pathway, which is very important for immune system regulation. We have signal molecule growth factors, 7-spanner receptors, which are important for sensing light, smells, serotonin. And then we have nuclear receptor proteins that are important for the signaling of our steroid and non-steroid molecules. And this is an important one that you'll learn more in biochemistry if you haven't taken that yet, or if you decide to take, let's say, a nutrition class, something like that. Okay. So first off, NF-kappa B, all right? NF-kappa B is a dimeric protein, meaning it has two parts, right? So we have to have two pieces of that protein. And this protein helps control transcription as well as cytokine production and cell survival. Now, cytokines are just small protein molecules that are involved in the immune system regulation. And ligands, which means it's just something that is a binding, okay, can cause our NF-kappa B to turn on more than 150 genes in the inflammatory system and the immune system, as well as during development in the cytoplasm. Now, I mentioned that the NF-kappa B is a dimer. That dimer is made up of the P50 and the P60 protein, therefore it is a heterodimer. So this might be 50, this would be 65, right? And then we have our heterodimer. So let's talk a little bit more about NF-kappa B. So, NF-kappa B, as I said, plays a large part in the immune system and so forth. Well, NF-kappa B, for one example, can be inhibited when the P50 subunit binds to a cancer suppressor protein. Okay? So if it's inhibited, therefore, it cannot help with transcription or increase transcription for the rest of its purposes. Okay? A second example of how NF-kappa B may be regulated, it can be inhibited by its binding to another protein called I-kappa B. So NF-kappa B is bound or can be bound and inhibited by the inhibitory protein I-kappa B. Well, what you can do to release this inhibition is phosphorylate I-kappa B. That will cause I-kappa B to release NF-kappa B. At this point, NF-kappa B can travel to the nucleus. Okay, it was originally in the cytoplasm. And at that point, once NF-kappa B is in the nucleus, it can go ahead and regulate any transcription as it normally would. At the same time, this phosphorylated I-kappa B that is no longer bound to NF-kappa B can be ubiquitinated. And ubiquitin, if we remember back to the protein unit, is just a small protein, and that can have many different outcomes, but in certain outcomes, when a protein is ubiquitinated, it is sent to the proteasome and degraded, so broken down, thrown into the trash. Okay, so these are two separate examples how NF-kappa B can be regulated, in which case it either is, if it's inhibited, it can't affect transcription. If it is not inhibited and able to act in the nucleus, then it can affect and be a regulatory protein. All right, next we have the JAK-STAT pathway. So this pathway is turned on by a cytokine, so for an example, interferon alpha. And that'll attach to the cell surface receptor. So first of all, let's write out what is JAK. JAK is a kinase, and it's called Janus kinase. Remember, kinases are responsible for phosphorylating things. And then we have STAT. STAT stands for signal transducer and activator of transcription. Okay, so the JAK-STAT pathway. We have just, if we were talking about the previous slide, okay, if we have our cell, okay, here's our nucleus. We have a receptor. Okay, we have something binding to that receptor. In this case, the cytokine is binding. Okay, JAKs, okay, the kinases come together. Those JAKs will activate the kinase, which phosphorylates pairs of our stats. Okay, those phosphorylated stat monomers will dimerize, so these stats are as monomers. Okay, they will be phosphorylated. We'll just say that it's a yellow piece. That causes them to dimerize. Okay. And they do that via a specific domain on the stat molecule called an SH2 domain. And those stat dimers will then travel we'll call the nucleus in purple over here. Okay, so that stat dimer will travel into the nucleus and bind a specific DNA sequence related to transcriptional enhancement. Okay, and so this once again shows you that a molecule from outside the cell can affect what happens inside the cell through a series of events that happens to, in this case, deal with the binding of a molecule to a receptor, a phosphorylation of other proteins, and those proteins being sent into the DNA, being sent into the nucleus to bind to DNA. Okay, and at that point, it will affect transcription. All right, another example of signal transduction would be our signal molecule growth factor. So a couple examples being epidermal growth factor, or EGF, as well as insulin. Remember, insulin is the protein that is sent out by the body, okay, by the pancreas, when in response to glucose in the blood, so blood sugar. Right? When you eat something, you have carbs; those get broken down, absorbed from the stomach through the intestine into the blood. Insulin's released. Insulin takes that glucose into the cells. Okay, so how this works is that we have a binding to the receptor, activating a dimeric, so a 2 protein. RTK, which is a receptor tyrosine kinase enzyme. Now, the dimerization requires the adapter proteins GRB2 and SOS. That's just some extra information for your own knowledge. So, we have this signal causing the RTK dimerization. Okay. That dimer binds and activates a protein called RAS, which will bind a GTP molecule. That RAS will then bind to and activate another protein called RAF. Now, RAF is what is called a MAP KKK, which is mitogen activated protein. Okay, we usually just call it MAP. It's a MAP kinase, meaning it phosphorylates a MAP kinase. All right, it's quite heavy in nomenclature, but we'll understand it as we go to the next step. So this RAF, which is our enzyme, a specific MAP kinase, RAF will then phosphorylate, okay, since it's a kinase, it will phosphorylate MEK. Now MEK is a MAP kinase. All right? Then MEK, once it's phosphorylated, will phosphorylate a MAP kinase. So a MAP K. Okay? And this MAP K is a different type of kinase than the one that we talked about up here. It is a kinase that is called a serine threonine kinase. So what are the differences between these kinases? Well, the RTKs, they phosphorylate a tyrosine amino acid. Okay. This one down here will phosphorylate proteins at certain amino acids of either a serine or a threonine residue. Okay. So now that MAP kinase is active, it is still a kinase. MAP kinase will phosphorylate a bunch of other transcription factors. And these transcription factors, now many of them by the phosphorylation, will be activated, and now they can go and bind to the DNA in the nucleus and enhance transcription. All right, so it's a very complex process, many steps, but the whole outcome is to affect the transcription happening in the nucleus. All right. Now, we have other ways of regulating transcription, utilizing certain proteins called 7-spanner receptors. Now, what is a 7-spanner receptor? All right, if this is the cell membrane, we have a protein that crosses the cell membrane 7 times. So 1, 2, 3, 4, 5, 6, 7. Okay, and maybe that's its N-terminal tail, and this is its C-terminal tail. Okay, and what we have here is it's crossed 7 times. 1, 2, 3, 4, 5, 6, 7. Okay, so that's what the 7-spanner receptor is. And that's just, there are several different 7-spanner receptors. So how do these work? All right, very similar in the fact that there's ligand binding that's activating a process. Okay, so specifically, we have ligand binding activating trimeric proteins called G proteins. So trimeric meaning 3, right? 3 different polypeptide chains. Okay, so each one of these subunits, we have an alpha subunit, a beta subunit, and a gamma subunit. Okay, so in order, how this happens, we have the ligand binding activating that protein. Okay, now when GTP binds our alpha subunit, that will activate adenylyl cyclase, which is an enzyme. And this requires GTPase activating protein, also known as GAP. Okay. So adenylyl cyclase normally converts ATP to cyclic AMP, and we talked about this in a previous lesson, this adenylyl cyclase enzyme. Now, the CAMP, we've also talked about this, will bind to protein kinase A to activate it. Now, protein kinase A, otherwise known as pKa, will then be able to phosphorylate many different proteins, so some of which can be transcription factors. So pKa, once activated, can phosphorylate transcription factors, okay, which will bind to certain pieces of DNA in the nucleus and affect transcription. Protein kinase A can also phosphorylate another protein called CREB, which will bind to a specific DNA sequence called CRE, as well as be bound by CBP and P300 protein, which is CBP stands for CREB binding protein. So CREB BP. And CBP P300, this protein, is normally considered a transcriptional activator. So it turns on the transcription of genes. Okay, so there are 2 different ways that protein kinase can affect gene expression. Okay. All right, let's talk about another example. We actually have 2 examples in this one. Let's talk about how we have 2 different diseases, cholera and whooping cough, otherwise known as pertussis. So cholera, all right? Cholera is caused by the action of the bacteria Vibrio cholerae. And this Vibrio cholerae produces a toxin called the cholera toxin. Now, how does this work? What's the mechanism of action? So the cholera toxin catalyzes an ADP ribosylation. Okay, so that's basically adding a ribose group to ADP together. Okay, and this happens via the use of our old friend NAD, nicotinamide adenine dinucleotide. We've talked about this one before, and it's useful in energy. Now, this ADP ribose will bind to the alpha subunit of the G protein, okay, as we talked about in the previous slide, which doesn't allow the GTPase activating protein gap to activate GTPase. So therefore, you can't convert GTP to GDP. All right, well, what does that even mean? Well, that means that we have constitutive activation of our adenylyl cyclase enzyme. Constitutive means it's always on. Okay? Now, if our adenylyl cyclase is always on, that means that we are continually turning ATP into CAMP. And if we have a bunch of CAMP, that's going to keep pKa, protein kinase A, constitutively active, right? And if pKa is always active, it's always going to be phosphorylating things. And in these certain cases, it will phosphorylate certain transcription factors to turn on gene expression and can lead to the types of diseases or the types of symptoms from cholera or whooping cough that are common. Okay. Now, whooping cough specifically is caused by the bacteria Bordetella pertussis, and that bacteria also produces a toxin called the pertussis toxin. Now, this pertussis toxin acts in a little different way than the cholera toxin does. So let's talk about this. We have pertussis toxin binding to a protein called G sub I. Now, G sub I is a G protein inhibitor. Okay? So normally, G sub I will bind to and inhibit adenylyl cyclase. Okay, so opposite of cholera. Okay, so cholera adenylyl cyclase is always on. G sub I normally turns adenylyl cyclase off. However, we have pertussis toxin binding pertussis toxin binding to that inhibitor, preventing that normal inhibition. Therefore, we end up with the same outcome. We have constitutive activation of adenylyl cyclase leading to continual conversion of ATP to cyclic AMP, meaning continual activation or constitutive activation of protein kinase A, leading to transcriptional regulation at increased rates. Okay, so these are just a couple ways how we can utilize the previous material in this unit to understand how a couple different bacteria create their symptoms that we see for medical purposes. So going on to a different type of transcriptional regulation, we have what are called nuclear receptor proteins. Okay, and these are proteins found within cells that are responsible for sensing both steroid and thyroid hormones, as well as certain other molecules. Okay, so first off, we have our steroid hormones. So, for example, our androgens and estrogens are sex hormones. Our glucocorticoids, which are involved in water retention. They're going to bind our cytoplasmic nuclear receptor protein homodimer. Okay, so it's a dimer of the same two things. Okay, two different polypeptides. So these steroid hormones and these will bind our NRP homodimer. This is what is called a type 1 complex. Okay, so the type 1 complex will head to the nucleus, bind to an inverted repeat DNA sequence called a hormone response element, or an HRE. Okay, at that point, once the HRE is bound, then transcription can be regulated either in a positive or negative manner. Okay. A second way that nuclear receptor proteins act is sensing hormones, such as vitamin A or thyroid hormone. They will bind to NRP heterodimers. So in this case, it's two different proteins, right? Okay. This is what's called a type 2 complex. And this type 2 complex will travel to or actually is already in the nucleus, and it will bind direct repeats in the nuclear DNA and then affect transcription regulation. Now, a third way to look at our NRPs and how they might affect transcription would be any unbound heterodimeric NRPs, so not bound by hormone. They can cause deacetylation of histone tails, the lysine residues on histone tails. All right? Now, these heterodimeric NRPs that are bound by a hormone, so such as these right here, can cause the acetylation of those tails. And remember, when a histone, when its tails are acetylated, that often, depending on the histone code, right? It's very complex, but it often is related to the loosening of chromatin, meaning transcriptional activation, whereas the deacetylation of histone tails usually means the compaction of chromatin, meaning decreased transcription and replication. Okay, so now let's move on to something that is also regulating transcription, but in a completely different way. And actually, this is a really, really cool mechanism. Okay, this is what's called RNA interference. And this is a method that has actually been adopted by many research labs, and I'll show you an example in a couple slides, to actually choose what genes get silenced or expressed. Okay, so this is coordinated by a complex called the RISC complex, the RNA-induced silencing complex. Okay, and it's a ribonucleoprotein complex, meaning it has both RNA and protein. Okay, and this complex has been most well studied for having a function in the degradation of a specific target mRNA. Okay, and if you target mRNA to be degraded, that will decrease the level of the transcripts available to be translated. So therefore, in essence, it is functioning to decrease gene expression of a specific gene. Right? So we have very important players. We have RNase enzymes. So an RNase is an enzyme that breaks down RNA molecules. So these RNase enzymes called dicer and Drosha trim double-stranded RNA to form either small interfering RNA, siRNA, or microRNA, miRNA. These RNAs then get incorporated into the RISC complex, leading to specific mRNA targeting and then degradation. Okay, so it targets to a specific mRNA and does not allow translation to take place. It's actually really, really cool. So this process we call RNA interference, or RNAi, and we find this in many eukaryotes, and it was thought to be developed as a very important mechanism of gene silencing and viral infection defense. So let's talk about the RISC pathway in a little bit of detail. So first of all, what is an activated RISC complex? Well, that composes the RISC ribonucleoprotein, as well as a microRNA or a small interfering RNA, and an ATP molecule. So how does this all go down? So first off, Dicerandrosia cleave a double-stranded RNA into short 21 to 23 base pair fragments with a 2-nucleotide 3' overhang. So simply, it looks like this, okay? Where this is 2 nucleotides. Oop. Okay, that is 2 nucleotides long. Okay, this is our 3', 5', 3', 5'. Okay, and then this whole thing is 21 to 23 base pairs. Okay, and this is double-stranded RNA. Okay. All right, so that's the first thing that happens. Then we bring in a different RNase called Argonaute, which is also termed slicer. Okay. So the RNase Argonaute then associates with single-stranded RNA, associates the single-stranded RNA with the RISC complex to act as a complementary strand to whatever the target mRNA is. Okay, so this double-stranded piece right here that's been been processed by Dicerandrosia can be separated into single strands, and that's what Argonaute's working with. Okay, now the complex will bind to our target mRNA, so whatever piece of gene material that you don't want to end up turning into a protein. So the complex binds to the target mRNA and silences it. Silencing meaning it's not being turned into a protein. Okay, no translation. All right, so there are two different ways this occurs. If we're talking about an miRNA being used, okay, microRNA. So the microRNA in the RISC complex binds to the 3' untranslated region. Okay, that's a part of an RNA. And it is untranslated, therefore it wouldn't have turned into protein anyway, but it is a great spot for regulatory proteins to bind and affect whether something gets translated or not. So miRNA in that RISC complex binds to the 3' UTR with a mismatch, and it will block transcription. Okay, the other way that we can affect translation, if we have a small interfering RNA in the RISC complex binding to the mRNA without a mismatch, and this will cause RISCs to cleave the mRNA. Okay, so cleaving it versus just blocking transcription. Okay. All right. Now, mRNA degradation happens at a certain spot in the cell, okay, called a P-body. So mRNA degradation's localized in P-bodies, which are found in cytoplasmic space and often stained darkly and under that you can look under a microscope. Okay. So importantly, we have activated RISC. So the RNA-induced silencing complex being implicated in formation of nuclear heterochromatin. So meaning we can have RISC actually not just cleaving these mRNAs, right, but can actually somehow be a part of epigenetic changes. So actually not even getting transcription available, right? Not even allowing transcription to happen. So that's kind of cool. So here's an example of the RISC pathway and actually how scientists have been able to utilize it. So what we would have is viral, right? If there's an infection, they make double-stranded RNA. You can break that into siRNA via Dicerandrosia. Those double-stranded RNAs can enter into the RISC complex. Argonaute can take a single-stranded RNA and utilize that either as an miRNA or an siRNA, right, to either degrade the mRNA or to affect possibly the epigenetic state of the heterochromatin. Now, what scientists can do, they can target individual genes and silence them. So here we see a wild type, so a normal petunia plant. And these two are lab-created petunias utilizing RNA interference, RNAi, using the RISC pathway. So what they've done is, through selective targeting of the mRNAs specific for the color, they have silenced them in the white portions. Okay, so this is pretty cool. And RNAi can be used other than just for cool, phenotypes. It can actually possibly be used for more functional things, such as something that we'll talk about a little later, maybe gene therapies, something similar to that. All right, so that's your transcriptional regulation. And now we'll have just a few slides, a few instances of translational regulation. So just like prokaryotes, eukaryotes mostly regulate transcriptionally. However, they can regulate translationally. So you can have global regulation of translation, meaning you regulate all translation, not specific genes, okay, by phosphorylating the protein EIF2 that we talked about before. This is eukaryotic initiation factor 2. Okay? If you phosphorylate EIF2, that protein can't bind GTP, and if you can't bind GTP, you're not able to bring the starting codon, right, the charged tRNA with the initiating methionine, you can't bring that to the ribosome because this EIF2 GTP complex is required to bring that to the 40S ribosome. So if you phosphorylate EIF2, you can't translate anything. Okay. Another way of global regulation, okay, so we have phosphorylation of EIF2. Another form of global regulation is competitive binding of the 5' cap of mRNA. So normally that mRNA, remember we have a cap, right? And that cap is normally bound by EIF4G. Okay? But phosphorylated regulatory proteins called 4E BPs, so binding proteins, will bind to, the cap is normally bound by EIF4E, which is then bound by EIF4G. Okay. But if these, let's see the cap again. If this EIF4E is bound by these 4E binding proteins, right? Then the EIF4E will not allow the binding of 4EBP will not allow EIF4G to bind, and therefore you will not allow translation to occur. So this can also be used to regulate specific mRNAs, but in general, if you affect the binding of initiation factors for translation, binding the actual mRNA, you can affect all mRNAs that are undergoing translation. So now we can talk about a couple different ways of translational regulation, and this is what's called translation-dependent regulation. So this is when translation has already started and it is in the elongation phase, okay? Now, we have two different types. We have non-sense mediated mRNA decay, and we have non-stop mediated mRNA decay. All right? So non-sense mediated mRNA decay is when you come across an mRNA that has a premature stop codon. Okay, so what you want to do is degrade those mRNAs so that you don't even have to utilize the energy and the amino acids and the tRNA, the whole process, to make this short protein, which will end up being degraded by the proteasome anyway. So you can degrade mRNAs with a premature stop codon by removing either the 5' cap or the 3' poly A tail of the mRNA, in which case you have now freed up either one of those ends to nucleolytic digestion. So exonucleases can now come in and just chew it up and degrade it, the mRNA. We have another thing called non-stop mediated mRNA decay, and this occurs when a ribosome is working on an mRNA that lacks a stop codon. Now, this can be pretty detrimental to the cell, because if there's no stop codon, basically the ribosome is stuck with that mRNA attached to it, as well as the protein coming out. Okay? And it's just stalled, and nothing can happen. You can't release the polypeptide, you can't separate the ribosome, and which means you can't then translate a new mRNA. So, without that stop codon, the ribosome is stalled. You can't do anything. So we want to be able to fix that. So this is rescuing ribosomes translating mRNAs without a stop. So if you don't have a stop codon, that poly A sequence in the tail gets translated, and that poly A, so the AAA codon, gives you lysine. And what this does, that causes the ribosome to stall, and it acts as a signal, okay, that there's something wrong. So what happens is we have eukaryotic releasing factors 1 and 3 binding to the ribosome. Okay, 1 and 3. Binding to the ribosome and dissociating the ribosome from the mRNA. Okay. At that point, the endonuclease, which actually is an unknown endonuclease as of this point, researchers are still delving into this information, but this endonuclease will degrade the mRNA from 3' to 5'. Okay, and the protein, if it isn't able to fold properly, which is very likely due to all of the extra lysines, will likely just be signaled to be degraded, be sent to the proteasome. Okay, so these are two different ways that you can have translational regulation actually once translation has occurred. All right. So, transcriptional regulation, translational regulation, and now on to epigenetics. All right. We touched over this a little bit when we talked about chromatin organization. I think it was unit 5. But we're going to talk a little bit about this again, all right? Give you a little more detail. So, epigenetics, what is it? That is inherited patterns of gene expression resulting from chromatin alteration. So, it's not an alteration of the DNA sequence that is heritable. So, it's not a change in the base sequence, A's, T's, G's, and C's, okay? But it is a heritable. So passing from generation to generation, it's a heritable trait of extra non-DNA related changes in terms of the base itself, but it could be due to just small changes, such as a methylation, such as acetylation, all right? And it can be on the DNA sequence as well as on the nucleosomes itself, all right? So, all right, so this can happen. We'll talk about 3 different ways. DNA methylation, the nucleosomes or the histone proteins, as well as something called polycomb repressors. And a very important point of epigenetics is that DNA sequence does not act alone in passing genetic information to the future generations. Meaning, the methylation state, let's say, of DNA or of histone proteins, right? And the methylation state usually has to do with what genes are silenced or active, right? That is able to be sent from generation to generation. So DNA methylation usually occurs at CPG sites or CG sites. The little p, that just says it's a C right next to a G, only separated by a phosphate, which is what's in the backbone of DNA. So DNA methylation occurs at our CPG sites via the DNA methyltransferase enzyme. So what you do is you methylate cytosine to get 5-methylcytosine, all right? And that can, keep that methylation even through the next generation. However, one drawback to this is that actually spontaneous deamination can turn this 5-methylcytosine into a thymine, right? Over time. Remember, this cytosine, even as a 5-methylcytosine, will base pair with a guanine. Well, if it gets deaminated, it now becomes a thymine, okay? And on the other strand that wasn't touched, it's a guanine, okay? Well, what you need to do is, we see that this isn't a proper DNA pair, so there's a DNA repair that needs to come on. Now, if this T is repaired back to a C, no harm, no foul. But if this T is not repaired, but the G is repaired to an A, now we have a difference in the original base sequence. So an epigenetic change has actually turned into a genetic change that gets passed on to the next generation. Okay? So this can cause DNA mutations if not properly repaired. Okay? So CPG islands, I just want to talk about because you'll hear that a lot, probably if you read some research papers or if your professor's talking about genetics and especially DNA methylation. CPG islands are just regions of DNA with a high frequency of CG sites, and they're usually about 200 base pairs in length, and they're associated with promoter regions of a lot of our mammalian genes. So usually when you see CPG islands, that is something that should bring to your mind, oh, we are in the promoter of a gene. Okay, not all the time, but very frequently. And methylation of many of these CPG sites will silence gene transcription, okay, leading to heterochromatinization, okay, the formation of heterochromatin. Now, the methylation pattern, as I've talked about before, can be passed on to daughter cells, and this methylation will prevent the binding of a transcription factor called SP1 to these CPG islands. And the binding of SP1 is part of transcriptional enhancement, okay? Now, we also have this other protein involved with DNA methylation called MECP2, and this is a binding protein of those methylated CPG islands. So it's a repressor protein that binds to those islands in DNA, and it will act as a transcriptional repressor. So it decreases transcription by recruiting histone deacetylases. Remember, if you deacetylate histones, that usually compacts our chromatin, meaning there's no room for transcriptional machinery to get in and go through making your mRNAs. Now, a phosphorylated MECP2 protein has a decreased affinity for methylated CPG sites, Okay, which leads, sorry about that, typo, and that'll lead to an increase in transcription. So, MECP2, repressor protein. Phosphorylate it, it won't bind, therefore it leads to transcriptional activation. SP1, transcription factor, that's an activator protein. So let's look at an example using MECP2. So we have a protein called BDNF, brain-derived neurotropic factor. It's a protein implicated in learning and memory plasticity. So basically being able to retain your memories, okay, and reorganize them. Okay, this BDNF is released when our dendrites, which is a neural cell, get depolarized, so activated. BDNF release can lead to a phosphorylation of MECP2, meaning it will now not be able to bind the DNA very well, meaning you have an increase in transcription. Okay. Now, on a related note, but different specifically from this BDNF, if you have a mutation in the gene for the MECP2 protein, which is also the MECP2 gene, this will cause a disease called Rett syndrome. All right, and this is a syndrome that has clinical manifestations very similar to autism, so it will affect the neural function. And in addition to the autism-like symptoms, you have stereotypical hand movements like hand wringing, or repeatedly putting your hands in your mouth. Okay, it's almost unconscious. Now, these are some slides that we've already seen in the chromatin organization lecture, but I wanted to just have them again so that we could reference them. So remember, we have a histone core. We have 8 histone proteins. We have 2 dimers of H2A, H2B, 1 tetramer of H3H4, 147 base pairs of DNA wound around, okay, and it's about 1.65 times around the core. We also have linker DNA, 20 to 60 base pairs, okay, and just that nucleosome will compact DNA about 6-fold. When you add in the linker histone H1, you can compact even more into your 30 nanometer fiber from your 10 nanometer, and that will compact DNA about 40-fold, and you can even compact even more than that utilizing scaffolding, so that you can make a bunch of loops. Now, we've talked about the histone code before. There are post-translational modifications of our N-terminal histone tails. That's part of the epigenetic code. If we remember, phosphorylation, usually adding a negative charge, right? Acetylation, positive charge, right? If you're adding a negative charge, that is going to decrease its interaction with DNA, so it'll loosen it, making it more transcriptionally active. Correct. Acetylation, right? Adding positive charge. Okay, and that's actually neutralizing the negative charge of lysine, which is actually going to open you up. Methylation will increase the interaction of DNA, meaning transcriptional repression. And then you also have ubiquitination, which can be binding sites for either transcriptional activators or repressors. We've talked about the histone code being very complicated, right? You can silence, you can have gene expression, gene silencing, you can have chromosome condensation, you can have DNA damage repair based on several slight changes in the epigenetic code, right, of the histone. We've talked about nucleosome assembly. When you replicate your DNA, you have to replicate your nucleosomes, and some of those nucleosomes get reused, right, in the next generation. And so what those nucleosomes, whatever they had for epigenetic changes, right, methylations, ubiquitinations, phosphorylation, acetylation, whatever, that will be transported, okay, with the nucleosome to the daughter cell, so that can propagate the epigenetic status or the status from one generation to the next. Okay, and what's very important are those parental H3, H4 tetramers. Okay. We've talked about the inheritance of the chromatin state. All right. Histone acetyltransferases can bind acetylated tails using its bromodomain and then therefore spread the acetylation, which would decrease the condensation of the heterochromatin, right, to make it more euchromatic and therefore transcriptionally active. Or you can have the same thing happening with histone methyltransferases, binding methylated regions via its chromodomain, causing a condensation and therefore more heterochromatization, i.e. transcriptional repression. Okay. So we'll just write this real quick. Histone acetyltransferase binds acetyl groups via its bromodomain. Okay, leading to a looser state, so which is more likely going to be a transcriptional activation. We have histone methyltransferase via its chromodomain binding methylated histone tails, which will tighten, right, condense, leading to a more likely repressed state. Chromatin remodeling, okay, is all part of epigenetics. As I said on the previous slide, when we have histone acetyltransferase as well as chromatin remodelers like the SWI/SNF complex, that allows you to loosen up the interaction of DNA with the histones and allow your transcriptional machinery to come in and therefore be in an activated state. If you have your histone deacetylases and your histone methyltransferases, that is going to cause a tighter association with DNA and therefore less room for your big RNA polymerase and all your initiation factors to come in and allow transcription. So this is a transcriptionally repressed state. Our last example, I believe, we have methylation of DNA and histones seeming to be correlated. So usually when both DNA and histone tails are methylated, this will be a transcriptionally repressed state. Okay, and acetylation of histones leads to the loosening of DNA around the histone, offering a higher likelihood of replication or transcription. So acetylation, higher likelihood of replication of and transcription. Methylation, transcriptionally repressed state. Okay. And finally, just as a mention, we have our polycomb repressors. All right. And these polycomb repressors just are a family of proteins that can remodel chromatin, okay, such that epigenetic silencing will take place. All right. PRC, that's just polycomb repressor complex or repressive complex. So PRC2 protein complex will bind DNA and trimethylate your histone 3 at lysine 27. Okay. And this will cause a repression. Okay, the PRC1 protein complex will also repress transcription. And then we have another protein complex called the MLL protein complex, which will reverse this repression by demethylating the H3K27. Now, a couple things I want to point out is that the PRC1 protein complex, okay, a mutation in this, okay, if you're a mutant, okay, like they can make in the labs, mutants die perinatally, meaning either at the time of birth or shortly after, no more than weeks, in under a couple weeks. Now, the PRC2 mutants, are what are called embryonic lethal, meaning they never get born in the first place. Okay, so these are really important complexes, and one more thing to show you how important they are is that overexpression, so if PRC1 and PRC2 are too active, right, there are too many around, they're repressing too much, if they're overexpressed, that is actually correlated with a more severe, okay, and more invasive types of cancer. Okay, so your cancer is likely to be you're worse off if you have overexpression of your polycomb repressor complexes. So that's the end of the lesson today. I hope you enjoyed our lesson, and I hope I see you back for our next one. So thank you for joining us at educator.com, and I hope to see you again.